Afternoon. So my topic today will be simplifying Decimet's membrane endothelial keratoplasty. So I am a very strong believer of evolution, and I like the words of this great Napoleon Bonaparte, who said, one must change one tactics every 10 years if one wishes to maintain one's superiority. This has been very true in case of in the evolution of endothelial keratoplasty, because there has been tremendous changes from penetrating keratoplasty to Decimet stripping endothelial keratoplasty to Decimet's membrane endothelial keratoplasty. So we have, in all endothelial diseases, we were replacing all the 5 layers of the cornea. In late 90s, Dr. Mellis and Dr. Price were the first people to think about why should we replace the entire cornea when only the 5th layer is having the problem. So they devised something known as Decimet stripping endothelial keratoplasty, which had only the stroma and the Decimets. So a year later, Mellis went one step ahead and he thought, why should the stroma also be done? Why can't only the Decimets can be replaced? And this led to the evolution of Decimet's membrane endothelial keratoplasty. So before we did the study, we did a literature search, and what was surprising was that there was no Asian study in the last 10 years for DMAC, and also the DMAC which came in 2006, hardly some 7 to 10 surgeons were performing this procedure. Why? Before we wanted to do it, we just wanted to find out, and we made a self-analysis, that these were the 2 questions which I had to answer. Do I know the orientation of the DM scroll? When you take the DM out, what happens? Is it scroll inside or scroll outside? We really didn't know. And no standard technique existed, like a cataract or any other surgery. Nothing was there. So this resulted in a lot of endothelial cell loss, and people were not doing. So the first study, what I did was, I had to take a lot of DMs and see what was happening. So the Decimets always scrolls on itself and exposing the endothelium out. So I'm showing you here a video. What happens, I'm just doing like a taco fold, putting the endothelium inside, I'm folding the Decimets, and see what happens when I leave it into the saline. The Decimets snaps open and the endothelium comes out. So this has been the major problem on DMAC, because when you start to handle the DM, and that's the problem. The second problem was there's no standardized technique. So we had to standardize. How will you prepare the DM? What was the injector to use? And some marking technique to know how the DM scrolls inside the eye. So this is just a small gist of video. I just want to show what we do. First I just marked with the trephine. I did not punch. And then with the Sinski I scored the Decimets. And then I stripped with the single pull technique. Put it 1/2 over the bad stroma. I punched with the 3 mm punch, and I make a mark on the Decimets, not on the endothelium. I close the punch, and then the DM is stripped. So this is the injector which we have made. It's a very simple one, a 1 cc syringe with a phaco tubing, the normal our IOL cartridge. It's done. You fill it with saline, and the injector is ready. And the Decimets which you have stripped, you can take it into the injector, you can see it, and then you put it into the eye which is already Decimet stripped, put one suture, do gentle taps. Once you see the mark, you close it. You don't need to take too much of time to see where the DM scrolls inside or outside. This is a very simple method. So after making a clear view, we did a prospective case study of 40 eyes of 39 patients during April 2015 to 16. And this is from Shankara Hospital, Coimbatore, consecutive cases. So we saw that the majority of the cases were pseudophakic bullous keratopathy. Fuchs were 20%, and bullous keratopathy was 10%, and ABK was 3%. Male preponderance was more. The visual acuity very well improved from pre-op 1.257 mean value to 0.265, which was statistically very significant. The core of our study was our endothelial cell difference. It was 327.95, correlating to mean loss of 15.1%. That was something which was really surprising for us also. We also had problems. We had 2 cases we had to reject. We have to repeat procedure. We had to do an air injection, and complication rate was 5%. We had 2 detachments, and the failure rate was 8%, and success was 92%. So now we have done cross more than 200, and this is the highest in the series in our country. And we have now started doing iridocorneal endothelial syndromes and all these cases which are really tough. And these are very simple cases, Fuchs, and also in graft failures in cases we try to do DMAC when stroma is very clear. So these are now, even these cases we try to do, because now after standardizing, the procedure has become very simple. So how come, as I told you before, our endothelial cell load was 15% when you compare to other groups which were 19%, more than like 22%. How could we achieve it? I can really say our outcome came because of these factors. Our technique of DM peeling had the shortest time. You keep peeling the DM for a very long time, you have problem, there is a lot of endothelial cell loss. Next, immediate utilization of graft. We are not like US or UK, somebody in eye bank prepares it in travels. No, we the surgeon do it, remove the tissue, and within 10 minutes inside the eye. Use a simple prototype injector, don't go for costly ones. We as Indians, we have to be, make in India, we have to make better ones because the other ones are too costly. And again, L marking technique, once you know how the DM really opens inside, the surgery is almost done. So what makes our study unique is the first study in the Indian population, and we followed a technique which was a standardized one. It is cost effective and does not need any costly instruments. Unlike DSAC, it had the least endothelial cell loss in 6 months period compared to any published DMAC study, stressing the need that if you standardize the technique, you will ensure success in all cases of DMAC. Thank you.
Dr. Siddharthan, that's a lovely study. You did it in 2015, almost 3 years ago. Have you published it?
No, see, what happened is this is one of the best paper. It got awarded in Tamil Nadu in 2016. So then it got selected into this. So by the time this 200 patients, what we have done, I have not, we have not published it yet. But this 40 patients, I have not published because it has serially gone into papers I was not supposed to present it. But now we have crossed 200 patients, and even probably we will be publishing very shortly.
Okay. Now you may have better data of endothelial.
Yes, sir. It is almost. Yes, sir. Now the 2 years means like now almost 1.5 years. It is definitely showing the same results only, sir. Meaning that the endothelial cell loss correlates to any other penetrating keratoplasty or DSAC. It does not show any this thing. But definitely when you compare with penetrating keratoplasty, the amount of endothelial cell loss is lesser, and it is better than even
this. Endothelial count is after 6 months. What was the period?
Yes, after 6 months, sir. After 6 months.
Dr. Siddharthan,
Yes,
congratulations on your work.
Yes, sir.
But what is new that you have done?
So the thing is that even now, sir, there is no standard technique to remove the DM. So what we have done is, we have done these procedures, and now I run a course where we trying to standardize these techniques, and whoever the students I've trained, they go back and they come and say, these steps, what you have taught us, what you have done.
No, that's exactly what I want to know. What is it that you have done differently from other workers?
Yes, sir. The difference is that there is no standardized technique till now it's available, sir. That is, as we say, you use a prototype injector, you use an L marking. Everybody keeps doing it.
Marking has been there.
Marking has been there, sir. But the problem is now because of the
Injectors have been there.
Everything has shown a lot of endothelial cell loss, sir. As I tell you, it is a combination of things. I'm not telling you the injector alone, our injector alone has made this change. What I'm telling you, if you can standardize each and every step, that is when you strip the DM, you concentrate that what best can be done. Because most of the studies, sir, as I told you, there is no Asian and Indian study. Most of the studies outside, they mostly have this pre-stripped tissues, already stripped, and it is already come. So we
Not
thought...
in India.
Yes, sir, not in India.
Not in India.
Yeah.
In India, it's always
The volume is
that's utilization.
Yes, sir. The techniques, as I told you, whoever has done, they have stopped doing DMAC. The process, it has evolved. Actually, I didn't mention this. 2013-14, we had done another 30 to 40 cases which had actually failed in 13-14. What we learned from that thing was that time there was no L marking. That time there was no proper how to strip. What was the force?
Or S marking,
Okay, whatever marking, there was no forceps to strip properly. There was no proper injury. We first used the ICL cartridge. So it evolved, sir. It evolved, and then what we found out is these things, when you standardize, when you don't keep altering. I use an ICL.
So when you got your first results,
Yes, sir.
at the end of 15 December is when you concluded the study. 2015 December.
That was the study period, sir. Then after that 6 months, so it's
Okay,
June.
You
Yeah.
results, middle of 16.
Yeah.
You had significant proof of having better results.
Yes, sir.
You considered it important to present rather than publish?
Sir, because I had in Tamil Nadu Ophthalmic Association, we had a very prestigious gold medal. So that paper, they say that you should not publish anywhere. So I gave more preference to Tamil Nadu Ophthalmic Association, sir, that you can, because that CP Gupta, it's below 40.
I'm just trying to see the fallacy in the system. I'm just trying to bring out a fallacy in the system. That means now if you're going to publish, you're going to lose a whole lot of advantage which you had in 2016 middle. So
Yes,
does
sir.
our system need evaluation?
That is the tragedy for most of the societies. They restrict
so-
to their journal, which may not have very high impact factors.
Yes,
That's
sir, actually, if I-
unfortunate.
Yes, sir. If
But
I had
I
have
think
published,
it's a smarter move to publish it,
Yes,
because
yes, ma'am.
that is forever. That
Yes.
becomes immortal rather than having a slot.
2015 you had a huge advantage. Thank you.
but the
Good
thing
work.
is, sir, even when I went now, still now there are no studies, so I am just going to do it once this is over. Sir. Thank you.
This is an excellent presentation. Congratulations.
Thank
I'm
you,
just
sir.
answering Suvan's question. This is a real disadvantage. You try to patent anything. If you have published it
I
anywhere,
have.
even presented,
Yes.
they will not accept it.
Yes, sir. But there
Even
is-
if it is came as a news item in a paper, you can't.
yes, I know. There is, because I have patents, so I know
That's
that.
what I'm saying.
I know that. But then we have to have a system by which, if he's done work and he's presenting his own work at a conference further down, let's say he has presented published his work, he submitted for publication, and then he's presenting the same thing in TNOA or MSA, whatever it is. He should get credit for his work. His work, that is what is called prior art in patent language. So it's my work, I'm presenting, that cannot go against me.
No, but they say it loses the novelty. The word novelty.
Novelty, yes, but that's prior art. It goes back to when I first published. It goes to published. So if I have published my work, I get credit for that dates back to when I first published. So that way publication is a better form of claiming first.
Scientific committee
Yes.
I understand that, sir, but as I wanted